Hey, hi guys. So we are going through the OCR paper, the AS paper 2017, and it's the paper 2 of this exam series. So just so that you know, I write the answers that I would have written. I then show you the mark scheme and the examiner's comments, and then I show you exactly where I did and didn't get the marks. I make a note of every single mark that I get so that you can see, so I number the marks so that you can see exactly where I've got them, and I put the tick exactly at the word that I think I've actually achieved the mark. So I'm hoping that that is helpful. But let's get started. So here we are on question 1. We've got drawings of 2 cells. One which has got an awful lot more detail than the other. Cell B was observed by an electron microscope. Give one piece of evidence from figure 1.1 to support this. So this isn't about you knowing about electron microscopes. This is about you identifying the evidence that you are given. So hopefully it is apparent that even though here you can see what looks like possibly a nucleus, and this looks like the permanent vacuole, we've got other structures here, but they're not clear. However, in this picture, we can suddenly see all of the detail of quite a number of the organelles in the picture. And therefore, the evidence is, notice that you can't just say that you can see the organelles. What you need to say is the names of the organelles, because of course the nucleus is an organelle. So does it make sense that you can't say, oh, you can see the organelles? Because of course you can see organelles in this one, you just can't identify them. So hence you can see and name some of the things that you can see. So there's the mark scheme. These are the points, these are the advisory points from the examiners, and as a result, I got the mark. Find one way that an image produced by a laser scanning confocal microscope differs from that from a light microscope. Now, the obvious thing to me, remember that laser scanning confocal microscope can actually see things and pick things up at different depths throughout the cell. And you visualize them because you use fluorescent tags in your specimen. So to me, it was the depth of the organelles that was the important thing. Notice that I haven't used the phrase, oh, you can see in 3D, because actually what you're seeing are definite layers. You're not seeing it in 3D like the scanning electron microscope. So here's the here are the possible marks that you could have gone, and I got actually I got mark point number 4 in the mark scheme here. I didn't number that one, I'm sorry. Identify the stage of meiosis 1 shown in the cell. Well, to me it is fairly obviously prophase, because I've just realised I've lost my little electronic pen, because the chromosomes are visible, and here we go, we're going to go straight into the 2 reasons for your answer. Notice here that you can't accept prophase 2 because the question has identified the topic of the question as meiosis 1. Based only on your observations, state 2 reasons for your answer. Well, for me, now, I was just going to say that I could see the chromosomes, or the chromosomes were visible. But I suddenly realized that that doesn't tell me that it's prophase. That just tells me that I can see mitosis going on. So that isn't good enough, which was why I went on to say that are randomly arranged, because in every other stage of mitosis, they're not randomly arranged. They're either, at the center or being drawn apart or at the poles of the cell. And the other thing that I thought, based on the opposite, state 2 reasons for your answer, and I nearly wrote for the second mark, oh, I can see the nuclear envelope. But I looked at this picture and I thought, in all honesty, I can't see the nuclear envelope. What I can see, that there is still a very distinct distinction between the nucleus and the cytoplasm. which is what I ended up writing. So I was a little bit careful in my wording here, trying really hard to make sure that I answered the question clearly. So mark scheme, examiner's advice. So I ended up getting mark point 1 and mark point 2. Sorry, in fact, I ended up getting mark point 3 as well. My goodness. Yes, of course, because I'd used the word randomly. So only a 2-mark question, I got the maximum. Another stage of meiosis is metaphase 1. Explain how the organization of homologous chromosomes during metaphase 1 increases genetic variation. So this is about those homologous chromosomes lining up on either side of the equator, independent assortment. But I'm feeling the need to explain what independent assortment actually means. So notice that I make separate points in separate sentences. I try and keep my sentences reasonably short because that tends to make your answer more clear. So homologous chromosomes line up as pairs on the equator. It is random as to which chromosome faces which pole. Notice that that is a really nice, easy way to say it is completely random as to whether they're arranged like this or arranged like this on either side of the equator. Which chromosome faces which pole? Good phrase there. That is independent assortment. Each chromosome of the pair will carry different alleles, because here I'm then thinking, what is it about homologous chromosomes that give rise to this genetic variation? Because of course, the maternal chromosome might have the dominant allele of a particular gene on it, and the paternal chromosome might have the recessive allele. So that is, notice that I haven't talked about independent segregation. Now here's a thing that just to be aware of. Metaphase 1 is independent assortment, the way the chromosomes line up. Independent segregation, that is the separation of the chromosomes as a result of independent assortment, and that happens at anaphase. Just to differentiate between those two phrases, those two terms, independent assortment and independent segregation. So here we go with the mark scheme and the examiner's notes, and therefore notice that they absolutely wanted for mark point 1, which I'm just putting in here, they absolutely wanted independent assortment, and they wouldn't have given the mark for independent segregation. Independent segregation doesn't happen during metaphase 1. And so I ended up getting 4 marks and therefore getting the maximum number. Use of stem cells is being evaluated for the treatment of certain human diseases. Name 2 potential sources of human stem cells, and for one source, describe an ethical issue associated with the use of the stem cells. So here am I sorting out and arranging my answer. Sources, embryo. Notice that the source is the embryo that gives rise to embryonic stem cells. And the source is the bone marrow, and that would give rise, or that would produce, a form of adult stem cells. And here is my ethical issue. Embryonic stem cells, of course, the producing of embryonic stem cells or the harvesting of embryonic stem cells might either damage or possibly kill the embryo. So you got a maximum of 2. So here's the mark scheme, here are the examiner's comments. You've got a maximum of 2 for stating the 2 sources. So I got those. I rightly identified one of the ethical issues for the embryonic stem cell, E1. Here are my marks. Notice the fourth mark down here was for saying that I included a statement indicating either a judgment, an opinion, or an understanding of this ethical issue. Now, I didn't do that because I'm going to be quite honest, I did exactly what the question asked me. I didn't expand this last bit of the answer. I got 3 marks out of 4. And to be quite honest, I'm just not, I know I would never ever have thought of writing anything like that last 4th mark, given the question that was asked. So I'm just not going to worry about it. Group of students wanted to observe the position of xylem cells in the leaf stalks of celery. Describe a procedure that they could use to do this. Well, this is the classic thing of letting coloured liquid go up the xylem of the celery by putting the celery into a beaker or cup of coloured water and then slicing the celery horizontally across the stalk and under a microscope. So this is what I put. Here's the mark scheme. Now, interestingly, I didn't realise, but the mark scheme was split into, you could either get marks for saying these 2 things or the marks for saying these 2 things. So as a result, and here are the examiner's notes, and as a result, I got mark point D1 and D2. I didn't give myself M2 because it was or, so I could either get either D or M marks. Now we get described 2 types of plant. We get described water starwort, which is a hydrophyte, like a water lily, that kind of plant, and a cactus, which of course is a xerophyte. And then it says, give one way in which you would expect the walls of the xylem of water starwort, the hydrophyte, and the cactus, the xerophyte, to be different from a herbaceous cotyledonous plant. That, in normal language, is a bog standard plant. One of those plants that has leaves that looks like that. Yeah, not the blade-like leaves like grasses. They're not dicotyledonous plants. So we're just talking about how would you compare, what in one way would you expect the walls of the xylem vessels to differ from a normal plant? So my answer was really brief. In the water starwort, because, and here we got a little clue in the question here, it's an aquatic plant which has its stems fully submerged in water. Of course, it's going to get lots of lovely support from the water around it. So there is less lignin in the xylem cell walls, or in the xylem vessels. Whereas in the cactus, there will be more lignin than a normal plant. So here is the mark scheme and the examiner's notes. So less lignin and more lignin. Now, interesting here, they really wanted to know that these answers were in the context of the fact the difference between these particular plants and a bog standard plant, as opposed to not a comparison between the species. Now, I'm going to be honest, I haven't made that clear, but I think according to the mark scheme, I believe that because these phrases are in brackets, they're not categorically required, I would be given the benefit of the doubt. I literally wrote this having taken on board the question, and I knew that this would have less lignin in than a normal plant, and this one would have more lignin. So I would have got the marks. Xylem and phloem, 2 vascular tissues found in plants, state 1 similarity and 2 differences between the structure, notice the structure of xylem and phloem. We're talking about how the leaves, I'm talking about how the cells are made, what adaptations they have. So this is what I put. Both are made up of adapted cylindrical cells. Different one, Xylem have no end walls, phloem sieve tubes have sieve plates, and Xylem cells are lignified, whereas the phloem isn't. Here is the mark scheme. I didn't get mark 0.1 because I didn't say that these cylindrical cells, I was kind of thinking in my mind, oh, this makes up a continuous tube, but I didn't actually say that the cylindrical cells were positioned or joined end to end. So I didn't give myself that mark. So I got mark 0.3 down here, and I got mark 0.1 about the lignin. Of course, there were many other comparisons that you could have made. Notice that for each of these differences, here's a really classic thing to remember in terms of how to answer these questions. If you're ever asked to compare two things or say the differences between two things, always say what both of them are like regarding one particular feature. So here I was talking about the end walls, no end walls in Xylem, sieve plates in phloem. Second difference, I was talking about lignin, yes in Xylem, no in phloem. Don't start muddling up your differences. Oh, Xylem cells have lignin, whereas phloem cells have sieve plates. Well, that isn't a difference. You've got to compare the same feature with both things that you're talking about the difference in. It's just like if you were saying the difference between me and somebody with dark hair. Carol's got blonde hair, so-and-so's got dark hair. You're talking about the feature about both things. Same feature about both things. Okay, bony fish and insects have different gas exchange systems. Both can be observed by dissection. Describe how you would carry out the dissection to display maximum detail of either gas exchange system. You can choose. It's not maximum detail of both gas exchange systems. It's one or the other, depending on what you have done as practicals in your school. So I'm going to choose fish. It's the one that I'm more familiar with. So remove the operculum, cut the gill arches, because this is what I would do. I would cut the gill arches from the body of the fish, and then I would add a load of water so that the filaments of those gills would be separate so that I could see them. Here's the mark scheme. There's the notes. So I think I only got one mark. I think I only gave myself one mark here because I wasn't sure. If you were talking about water, they wanted you to say that you observed them underwater. Now, I'm not sure that my adding water would have been quite enough. So on the benefit of the doubt, I prefer not to give myself that, just in case. Right, I'm not going to read out all of this because actually this whole description does not help you with any of the future questions. Let's just say they're talking about some beetles that carry air bubbles, water beetles that carry air bubbles underwater. And they did an experiment where they put 3 different beetles that do this funny thing of carrying an air bubble underwater. They put them in different temperatures, 10 degrees, 20 degrees, and 30 degrees, and they counted the number of times that these little water beetles came up to the surface to collect another air bubble. Of course, these beetles are insects, so they're using their spiracles, they're using that insect method of gas exchange, spiracles, in order to do their gas exchange. So here's your data, 10, 20, 30. Here's the number of times that the beetle came to the surface. Student made an error in their working. Put a ring round the error in table 3 and write the correct answer next to it and use the space below for working. So I did a quick bit of calculations on my calculator and realized that actually, because this, and look, it's recorded to the nearest whole number. There's the critical thing. This actually came out at, I think it came out at 19.131313. Oh, sorry, no, it isn't. It's 19.3 recurring. Sorry, my memory's wrong. And therefore, the rounding up was incorrect. It should have been rounded down to 19, not rounded up to 20. So I got the 2 marks here. In fact, if I'm honest, because I got the right answer, I didn't need the mark. I didn't need the working. Here's the gas exchange system of an insect. Name the structure labeled A. Well, they label this one for you. This is the trachea. So these are tracheoles. A is a tracheole, which was the right answer. Describe how the trachea of a mammal is different from the trachea shown in figure 3. So here we are. We've got to make sure that we are making it clear we have comparative statements about these 2 tracheae. So the trachea of a mammal is much larger. It contains cartilage in its structure, as well as having ciliated epithelium and goblet cells. And notice here, I've kind of said, up until the end of that sentence, I remember thinking, oh, I finished my answer. And then I suddenly realized I haven't spelt out that the insect trachea has none of these things, which is why I then wrote the following sentence. Okay, so notice that if you're doing a difference, you're doing a comparison, you have to say what the feature is like in both things. So here is the mark scheme. Here are the possible mark points. Notice the statements must be comparative. Now, I didn't get the first one for saying that it is larger. Look, I could have said larger diameter, but I didn't say that. I just said it was much bigger. Ignore bigger, wasn't enough to get the mark. So notice that I've only earned myself marks where I've actually finished making the comparative statement. So here I got mark 0.2 and here I got mark 0.7. So I got both marks. Alveoli are located in the lungs of mammals. Explain how alveoli are adapted for efficient gas exchange. Really important here to read the question carefully. We are not answering the question, explain how lungs are adapted for gas exchange. We are explaining, and notice that, let me finish that sentence. We're explaining how alveoli are adapted. We're looking right down at those individual air sacs at the end of those, that whole structure of the lungs. And we have to explain. Now, almost certainly, we're going to have to do some describing. We're going to have to say what the alveolus is like, but we are then going to have to explain how that makes it efficient for gas exchange. Okay, so explaining is about saying why or how it works. Describing is just about saying what something is like. So hopefully I'm now going to, now here is my tip. So I always teach this little equation. It has the posh name of Fick's law, and I've just put it in abbreviations here because this is diffusion rate is proportional to the surface area of the exchange surface, the concentration gradient, and it's inversely proportional to the diffusion pathway. In other words, if this were, well, this is pretty much a mathematical formula. If either of these numbers gets larger, the surface area gets larger, or the concentration gradient steepens, in other words, the gradient becomes a larger number, the diffusion rate will increase. If, however, the diffusion pathway increases, with this number on the bottom, the number we're dividing by, if that number gets bigger, does it make sense that the diffusion rate will decrease? So we want big numbers here, small number here. This is absolutely my way of thinking about how gas exchange surfaces, any gas exchange surface, is adapted to make it efficient for gas exchange. Because in every gas exchange surface, there'll be a way in which the surface area is increased, the way in which the concentration gradient is increased, and the way in which the diffusion pathway is decreased. So here am I thinking about each of these things with regards to the alveolus. So, alveoli, very small structures. Because of that, missed out that word, because that enables many of them to be in the lungs, in that volume, this increases the surface area for diffusion massively. So I've just said it increases the surface area, which increases diffusion. The alveolar epithelium is 1 cell thick. This contributes to a small diffusion pathway. I've said contributes because I'm being accurate, because of course you've got the alveoli, alveolus epithelium, and the capillary endothelium that the oxygen molecules and the carbon dioxide molecules need to diffuse through. So both epithelia are 1 cell thick. Called the alveolus, the epithelium. The correct name for the capillary epithelium is the endothelium. Sorry. So it contributes to a short diffusion pathway. Each alveolus is surrounded by elastic tissue, because here am I thinking about the adaptations of the alveoli or an alveolus. I did not include anything about blood flow here because I was very scared that that would not be seen as an adaptation of an alveolus. So I'm sticking to alveolus. Each alveolus is surrounded by elastic tissue, which enables efficient removal of air, because that elastic tissue stretches and then recoils. So this increases the concentration gradient of oxygen and CO2 between the blood and the alveolus. Increased surface area, so here am I absolutely nailing how each of these things increases the efficiency of gas exchange. Increased surface area and concentration gradient and reduced diffusion pathway is proportional to increased diffusion rate. So I've nailed why surface area, concentration gradient, and diffusion pathway are important. So it's a 6 mark, one of these 6 marker where it's leveled. In summary, science content determines the level, and the communication statement determines the mark within the level. So I'm going to jump. Oh, it seems like I've missed a slight bit off the mark scheme. I'm really sorry. So we need identification of more than, let's say, I can't remember, I think it's more than 2 adaptations of the alveolus, and I need a scientific explanation of more than 1 adaptation. So for the 5 to 6 marks, we need a identification of more than 1 adaptation and an explanation of more than 1 adaptation. Now, I have done that, and there is a well-developed line of reasoning which is clear and logically structured. The information presented is relevant. And I think that that is also the case. So I'm going to give myself 6 marks. You can go through all of these. Now, I thought it was very interesting because they talked about good blood supply here, and they talked about the fact that the alveolus was ventilated. I'm going to be really honest, to me, that is not an adaptation of the alveolus. I was absolutely thinking about structural adaptations. So yes, but anyway. Lymphocytes. Do feel free to ask me any questions if you have any. Really happy to do so. I'm doing this, going through this reasonably quickly. For those of you watching this who are not in live stream land, please do take the opportunity to pause the video so that you can have a go at the questions and think about what you would have written. Lymphocytes play an important part, form an important part in the specific immune system in humans, classified into B and T lymphocytes. Use ticks and crosses to identify whether the statement is true for B lymphocytes, belongs to B lymphocytes, T lymphocytes, or both. First one has been done. So, forms part of the immune response. Absolutely, ticks for both of them. Differentiates into memory cells. Both of them differentiate into memory cells. Produces chemical that causes lysis of an infected cell. Only true of T lymphocytes. B lymphocytes don't do that. And form plasma cell clones. That's something that B lymphocytes do, but the T lymphocytes don't. So here am I following the instructions of the question, and that's what I have put. Here's the mark scheme. So I got all 4 marks. I haven't included the examiner's notes on this occasion because all the notes were about, well, what happened if people had left things blank? All right. And I'm just going to say, if they tell you to do something in a question, do as you're told. Figure 4 shows the concentration of antibodies in a patient's bloodstream following a flu vaccination and then a subsequent infection with the flu virus. Describe where the primary and secondary immune responses are taking place and explain the differences. Not just state, not describe the differences, then you'd just be saying them. Explain the differences. That means you have actually got to describe the difference and then say why. It's a longer answer. Here we go. So I'm saying here how I am abbreviating primary and secondary. Primary, I've got fed up of writing it down here and I realized I could have saved myself a bit of time. Primary response occurs after vaccination only. So it doesn't occur here, it only occurs here. And the secondary response occurs after infection only. So I'm just trying to make it really clear. The secondary response is much faster than the primary because, here we go, so I've noticed that I've described a difference. It's faster than the primary response. And now I'm going on to explain it. As soon as I use the word because, or as a result of, or due to, or so, all of those kinds of words, nearly always after those words, you are then explaining. Because memory B cells turn into plasma cells in secondary, whereas in primary, clonal expansion takes place. I was thinking, you know, to be honest, primary response, specific immune response, is a multi-stage process. Really is. The T cells have to be activated and then clonally expand. The B cells have to be activated, clonally expand. And then the plasma cells of the B cells have to produce antibodies. So here is the mark scheme. Here are the notes. So I ended up with these 3 marks. In West Africa, during 2015, there was a serious outbreak of the Ebola virus, which spread by contact with infected bodily fluids. And you are then given 5 bullet points of things that were happening at the start of the outbreak and that were happening during the outbreak. And they were all factors that increased the spread of the disease. A number of common factors affect the spread of communicable diseases in humans, and some of them are relevant to the spread of Ebola. From the information above, discuss these factors and suggest what actions could be put in place to address them, or could have been put in place to address them. So discussing is not just quoting what has been said in these 5 bullet points. Discussing is giving a little bit more biological information that you will know from your biology course, and then suggesting actions for these issues. So this, I think, this certainly felt to me like the hardest question on the paper, because I wasn't sure quite what was required in terms of discuss, but as soon as you see the word discuss, think terminology of communicable diseases. Talk about pathogens, talk about infection, talk about how things are spread. We've been told infected bodily fluids. Now, that includes saliva, it includes droplet transmission, it includes blood-to-blood contact, it includes sexual contact. So here is my answer, which I've written in finer print because I realised I could have actually, look, you can see down here, I could have written more because I didn't get to address all of these points. In an exam, can I just say, my writing would be relatively much, I would have got many more words onto a proper exam paper than I have been able to do here, which is why I have run over the amount of space. But worth noting that, I would have written more for this question and possibly used up too much time. It would have been a tough call. So what my tactic was to go through each of these points, try and discuss it a bit, try and bring in a bit more of my biology knowledge, and then write an action for each thing. Again, this is one of those 6 markers. Science content determines the level, the communication statement determines the mark within the level. Here is the basic mark scheme, in as much as what are they looking for? So for 5 or 6 marks, they are looking for discussion, expanding on the bullet points for more than one correct factor, and a plausible suggestion of an action. Now, this is tricky, given that probably nobody, certainly before the pandemic, would have really have had any real experience or possibly even background knowledge of this kind of epidemic happening. This is the 3 or 4 mark one. Notice that you could either have correctly discussed one correct factor and or a plausible action, or you could discuss more than one factor or more than one action. This is a complicated mark scheme to get your head around. Finally, here are all the things, the kinds of things that they were looking for. This isn't absolutely exhaustive, but let's face it, this is what examiners would have used as a guideline. So given that this is, we've really got to be able to identify the discussion, an appropriate discussion of a factor, which I have designated F1, F2, F3, and the actions A1, A2, A3. I hope that all makes sense. I can now, notice I'm not giving myself marks, but I can kind of identify which I have covered. So as a result of the fact that I think I have appropriately discussed more than one factor and a plausible suggestion of one of those factors that I've talked about, I've got an F1, an F3, and I've got an A1, I'm up in the level 3 bracket. I think I would have got the 6 marks, but I don't want to be too big-headed about it. But hopefully that has helped, because I think this is a really tricky one to mark. And notice that if I was in the exam, I would have wanted to have tried to cover as many of those factors as I could, whereas actually you didn't need to cover them all at all, this was really about reading the question and understanding what discuss means, and the fact that they wanted actions too. This was more about reading the question and bringing in that knowledge from your A level than it was about actually the quantity of your answer. I hope that kind of makes sense. Okay, quick plug, just to let you know that I do have a website where I have got videos that cover the whole of the A-level syllabus, specifically for OCR A syllabus and the AQA syllabus. If you need to, if you'd like to have access to just the AS videos, you can subscribe to them for £50 a year. It's about one hours of private tuition. If you need both year one and year two, because you're in year two of your A level, that's £100 a year. Obviously about, depending on where you live, 2 or 3 hours of private tuition. If money in the family there isn't a lot of it, if you can come up with the money, you can earn yourself 10% commission on any of your friends that end up buying buying one of the packages. So an option to actually earn your money back or possibly make a profit. And there's also a freebie, so if nothing else, go and have a look, worth doing. Here we go. Study was carried out on butterflies in 2 different locations in the north of England. The 2 habitats were farmland and deciduous woodland. Deciduous means that the trees were not evergreen and lose their leaves in winter. Surveys were completed in 1992 and at 4-year intervals. Data were collected by butterfly transect sites on both habitats. Total butterfly population was estimated. In 2012, the general population of both of these habitats reached historical lows because of prolonged cold and wet weather. Between 1992, in other words, between the whole length of the study, or this graph particularly, the woodland had become overgrown due to a lack of active management. So the woodland became overgrown. In particular, the number of open spaces in the woodland had decreased. That might well explain why this dropped so low. Calculate the total percentage decrease in the number of butterflies on farmland between 1992 and 2012. Well, we're looking at this line up here. Starting point was 110. The ending point was 79. Now, we're doing percentage decrease. We're going from a higher number, we're stepping down to a lower number. We want to know what that decrease is. We want to know what the decrease is, which would be this kind of bit here, the difference between them. But we want that as a percentage of the original number. It's the percentage of the original. And that's why this is the correct fraction. 110 minus 79, in other words, the difference over 110. That's the correct fraction. Percentage is just a posh fraction. You times the fraction by 100. And that gives you 28.18. But it tells you to give your number, your answer to the nearest whole number. It's 28%. Notice how reading questions carefully, taking a deep breath and just reading questions carefully can stop you losing marks. Okay, so I got the 2 marks there. Using the data given in figure 5, compare the changes in the number of butterflies on farmland and woodland between 1992 and 2012. It's a 2-mark question. We're looking for 2 comparisons to being made. Notice it's comparisons. You need to be using comparison words. Bigger, faster, slower, whatever. So The number of butterflies decreased faster in woodland. Notice the slope is steeper, and it also decreased to a lower number than in the farmland. I didn't have to say that in the farmland because there were only two things. So as a result, so notice must be comparative statements up here. Oh, lost my, anyway. So I get mark point 2, I get the 2 marks there. Both habitats experience the same weather conditions. Suggest a reason for the difference in the rates of decline in butterfly numbers in farmland and in woodland. Why could there be a difference in the rates of decline? In the woodland, they declined more quickly than in the farmland. So I'm just kind of picturing in my mind, you know, a farmer's field with a in it and a load of woodland next to it. I'm just kind of going, what else? What could be different between those two things? And my immediate thing was that in the woodland, there could be more predators of the butterflies. So loads of different possible answers. Must specify which population is being talked about, rather obviously. Hope that makes sense. So I got the mark. Student made a following statement. These data show that the change in butterfly numbers was caused by changes in weather conditions in England. Comment on the validity of this statement. Right. Here, I would, if we're commenting on validity, I would go back to, personally, I would go back to the methodology that you have been told about. So I'm noticing that the only time that the weather conditions have been actually noticed here is in 2012. So I'm kind of going, well, they've only ascribed very low numbers to 1 year. And also, notice nearly always in these kinds of questions, when they give you a statement, the statement is nearly always broader than either the methodology that produced the data actually investigated. So here, weather conditions in England, well, this happened in the north of England. Is the weather in the north of England the same across the whole of England? No, it's not. And here's another really good word to use. So this, you know, here I give it, hopefully giving you some hints on great words that are useful. So I said not valid because the decrease is due to weather only applies to 2012, but also the weather in the north of England is not representative of the whole country. So what you could have said, the weather in the north of England is not, or the weather in the whole of England is not the same as the weather in the north of England. That would have been saying the same thing. But notice that the weather in the north of England doesn't represent the weather across the whole country. Representative is a really useful word to be able to use correctly, because it's often the word that gets you the mark, or that helps get you the mark. So I get mark 0.1 and I get mark 0.5. State one variable that scientists should control when carrying out surveys like this. They're looking for butterflies. What things should they keep the same? Well, the most obvious thing to me, so here am I kind of trying to picture the scene of this investigation. The most obvious thing to me is that they have to do the investigation when the butterflies are out and about. So don't do it in flipping winter, you won't find any butterflies. So therefore I wrote the time of year. So notice that you could have written, you know, any number of other things here, but this one to me was the most obvious. Butterfly species are in severe decline on farmland, include all of these types of butterfly. The ones in severe decline in woodland include all of these types. More types in severe decline in the woodland than there are in the farmland. State which habitat you would expect to have a greater species richness. So that is number of different species in the habitat, and give 2 reasons for your answer. This is a bit tricky because you could go round in circles on this. Because you could kind of go, well, if there are less in severe decline in the farmland, quite possibly there are more butterflies left. Or you could kind of go down the route, well, in the woodland, if they've identified all of these butterflies, even if they're in severe decline, that possibly implies that there were more to start with. Here's the, oh, sorry, I've just realized I've got this wrong way around. So this is what I put. I went down the route of it's woodland because more species were identified, even if in decline, and so more likely to be there originally. There was my logic, but I wasn't sure. This was a little bit of an unnerving question. And then I tried to think about something else, which, let's be frank, was a bit unrelated. But woodland would have more food sources. It's a classic thing that increases diversity, more food sources and more habitat. 2 classic things that increase diversity in an area. So I ended up with, I actually ended up earning 3 marks. I really felt like I really did have to explain. I've lost my little cursor. Anyway, really did have to explain mark point 1, and in the end I got mark point 2. So now we go on to a different type of butterfly. They're normally orange-brown. However, a deep olive green colour can be seen in some females, largely in the south of England. What term is given to this type of biodiversity? Well, we've got 3 types of biodiversity that you can choose from. Habitat biodiversity, genetic biodiversity, and species biodiversity. It's not habitat because it's not talking about the habitat. It's not species because we're not talking about lots of different species, because we're only talking about a single species. And therefore it must be genetic diversity by a process of elimination, which it was. Sorry, I've got this. Sorry, no, forget it. Give 1 possible benefit to the species of this type of biodiversity. I could have totally gone down the route of, why is it useful for it to have loads of different alleles in a population? Well, if a new disease comes along, less are likely to, not all of them will be killed off. And I just said, because I kind of thought it is primarily talking about this type of biodiversity, but I just thought, if I can, I want to talk about the colour difference, because then I know they can't stop me getting a mark for not applying it to the particular example in hand. So this is what I put. Less predation, as some at least will be better camouflaged. Either the green ones or the orange-brown ones will be, depending on the habitat, obviously. So here's the mark scheme. Here are the examiner's notes. Look, you could do the whole species survival when a new disease introduced. You could have said that. I got the mark. Only got a few more minutes left. Group of scientists decided to investigate glucose content of 3 types of fruit juice. They carried out Benedict's test on known concentrations of glucose and used these to calibrate a colorimeter, not a calorimeter. Calorimeter, completely different, measures the energy in food. Colorimeter measures the colour. Here's the results of their calibration. Plot a graph. Right, can I just say, this was an absolute nightmare to do on a PowerPoint size when it isn't whole screen. So I'm pleased. So this is what I would have done. Linear scales using the correct axes. The independent variable is along the x-axis. The mean absorbance is on the y-axis. Try to plot the points as accurately as I could. And here was the mark scheme. And here are the the guidelines. So notice, to get the axes the right way round and include the appropriate label and units got you the first mark. Points plotted correctly, and look, I was allowed within half plus or minus half a small square, so I think I managed that. And the line of best fit. Notice the line of best fit does not extend beyond the initial and final plots. Really important. Line of best fit. Quick way to decide a line of best fit can either be a straight line or a curve in biology. The aim of the line of best fit is to clearly show the trend of the data. But that line needs to represent all the data points. It is the most arty-farty thing you will do in biology. Notice that my line, so here's a guideline. If you draw a line of best fit, you want approximately equal numbers of points on either side of the line, as I've done here. I think there's about 3 on each side. Also, here's the way to refine your line of best fit. And notice this is true of a curve or a straight line. You actually want the distance between the points on one side and the distance between the points on the other side to be approximately equal. A line of best fit should never try and go through each point. All right, it is either a smooth curve, or it's a straight line. It's showing the accurate trend of the data, equal points either side of the line. If you can manage it at all, possibly as much as you can, equal distances between the points and the line. Hopefully that makes sense. Okay, students were provided with 3 different fruit juices. The Benedict's test was carried out on each fruit juice and samples were prepared for the colorimeter. Explain how the students would use the calibration curve to estimate the glucose concentration in the fruit juices. Here's what I put. Measure the percentage absorber and then use the calibration curve to read off the estimated glucose concentration from the x-axis. This was the mark scheme. Now, here's a point where, because I wasn't sure how to explain this, how do you read off a calibration curve? Notice what they have written in the mark scheme here. To find the concentration that corresponds to the absorbance. Really good phrase. I'm going to try and remember that phrase for OCR exams. So find the concentration that corresponds to the absorbance using the calibration curve. So that's what I'm going to use in future. I only got 1 mark out of the 2. Students wrote the following hypothesis. The higher the concentration of glucose in the fruit juice, the sweeter it will be. How could you carry out a controlled experiment, there's a clue in the question, to test this hypothesis without using a colorimeter? You've got to assess the glucose concentration by another method. The only other method that I could think of was using the semi-quantitative method of testing for reducing sugars, using Benedict's reagent. And then I said, get a single person to judge the sweetness by taste of each of the juices. You don't want different people doing each juice, one person, using an arbitrary scale of 0 to 10. 0, no sweetness, 10, very sweet. And see if the glucose concentration is proportional to the sweetness score. Now, I added this on because I went back and I wasn't really happy with that answer, I'll be honest. And then I haven't actually addressed the important word, controlled experiment. That means actually listing control variables. So here's me trying to couple of control variables. Volume of juice tasted, If you taste a whole load, you're going to get more sweetness in your mouth than if you taste a smaller volume. And 1 person to test all the juices. Here's the mark scheme. Here are the examiner's comments. And so these are the marks that I think I would have got. Taste the fruit juices to see how sweet they are, mark 0.1. Now, I think I would have possibly got mark 0.2. I don't know whether am I okay saying semi-quantify? Because here it talks about accept semi-quantitative test for reducing sugar. I talked about reducing sugar. I talked about semi-quantifying the glucose concentration. I think that would have been enough. I think that this is a separate point to these 2 lines here. Benedict's tests on each fruit juice and weigh mass of precipitate. Or you could say semi-quantitative test. I think I've got away with it here. I'm hoping so. Obtain rank order for sweetness. Well, because I indicated how you could grade the sweetness, clearly you could rank. I think that would have been enough. And I mentioned control variables. I'm going to be honest. I'd have got either 2 or 4. I think I'd have got 4. Suggest one reason why the results of this experiment might not support the student's hypothesis. Classic answer here, because taste is subjective. What colour we see things are subjective. How we taste things are subjective. How we hear things is subjective. Everybody sees, hears and tastes things slightly differently. It's not objective. It can't be given a number that will always be the same. So subjective, another really useful word to know how to use in exams. So got the mark there. And finally, going to finish just in time. Are both molecules transported in the blood that may need monitoring in people with different medical conditions. Here's a cholesterol molecule. State 2 ways in which the molecular structure of cholesterol is similar to the molecular structure of glucose. So here we go. 2 things. Both only contain carbon, hydrogen, and oxygen atoms, and both have at least 1 OH group. Interestingly, here was the mark scheme. Now, also, this is a list question. You're only allowed to state 2 things. I absolutely assumed that this would be one thing, the atoms that were in both molecules. However, it got separated into 2 mark points here. So I actually ended up earning all 3 marks. But notice only the first 2 answers would be given. So I got my 2 marks then. Glucose is if I had written something that was incorrect, it is quite possible that they would have taken away one of these marks that I had earned. It's really important that when they say put 2 ways, that you honestly only try and do 2 ways. I honestly did. Fortunately, this last mark here was also correct, so nothing would be taken away. Glucose, an important biological molecule required by cells for cellular registration. State the physical property of glucose that allows it to be easily transported in the bloodstream, and that is because it is water soluble. Things are either water soluble or lipid soluble. Glucose, water soluble. So here we go. And that is the end of OCR 2017 paper 2. Really hoping that has been useful, and I will see you again next Tuesday to go through another paper. Okay, see you soon. Bye-bye.